Hi there, my name is Robert Purrier, and I'm the Chief Scientific Officer for Imtact USA. We are an HPLC column company, and today we're going to be talking about ADCs, antibody-drug conjugates. This is such a wonderful science, to be able to direct delivery of a small molecule medicine, whether it be for cancer or any number of fields. This is such an important field of study. But the chromatography can be quite challenging. These are very large molecules. And today we're going to talk about what the issues are and how we make some columns that might help address this. So stay tuned.
All right, so let's talk about ADCs. I'm really so excited about this field of study. It has so much potential, and I also feel like we are uniquely suited to support the chromatography work that's being done in this field. So I appreciate you taking a few minutes to watch this presentation. Um, I'd like to start with just introducing us, just in case you've never heard of Imtact before. We're based out of Kyoto, Japan. It was started by a gentleman by the name of Itaru Yazawa. We call him Yazawa-san. He's really our founder, and he has such a wide background. He was an analytical chemist. He developed stationary phases at YMC. He even worked at Shimadzu on instrument side. So he likes to say that he really understands chromatography from all angles. And his passion and really his frustration with the available columns when he was a chromatographer um, was such a frustration that there really just wasn't a lot of good solutions for many of the analyses that he saw. And so he decided to start Imtact in 2000. And his passion is to, in his lifetime, to make the best possible columns that are really good tools for chromatographers out there. And that's the passion behind our company. We've got a lot of columns, and if you ever, if you see something like this, it might be a little daunting. Like, what are all these columns? There are a bunch of C18s here. I don't know what's what. Please reach out to me. I can help guide you. I know the subtle differences between each one and make your best recommendation for whatever you're working on. But for ADCs, we're going to concentrate on these three products here. These are what I see, over the last 10 years. These are the columns that the majority of our ADC groups are using, and we'll talk about why that is. Let me just further introduce these columns. So obviously, with ADCs, we're talking large molecule. So we're starting on the large molecule branch of this column selectivity chart. And under the size exclusion, the first split there, we have a column called the Entrada SEC. This is a high resolution silica-based SEC column. And really its highlights are that it can be used in either GPC or GFC mode, depending on what mobile phases you run through it. So very wide solvent usage ability on this column. But probably the thing that's the most important here is that it's designed from the beginning to be compatible with mass spec. So we can get that high resolution SEC MS work with this column very well. But the other main branch of this large molecule tree is the reverse phase branch. And what we see in reverse phase for large molecule work is almost everybody's using a wide pore C4 or something, either 300 angstrom pore size or 450 angstrom pore size. This is your typical protein column. We have one of those too. It's called WPRP. That's the first one listed here. But really, and hopefully I can show this a little later on, it really is not suited for ADC work. You get good high resolution with that type of column for proteins or other large molecules up to about 70 kilodaltons. When you're above that, it really loses its efficiency quite quickly. Now, that's where Presto FFC18 comes in. We'll talk about why that's the case. This is a non-porous column. but it gives a very high resolution it really opens up that very large molecule space up to high resolution reverse phase. And we'll talk about that in much more detail. The third column here is the cadenza HSC18. And why is this a big deal for ADC work? Well, this is a very special C18. It has a protein excluding ligand. Obviously, we'll get into more detail on that. But the big story here is you can directly inject your protein mixture, and the proteins will all wash out very early in the run while it's still under high aqueous, and they're not going to denature, but they're out of the column, but it's going to still retain your small molecules. So this is really a fantastic column. All right, so let's get into ADCs. What are ADCs? Well, I love this topic. I'm a scientist too, and this topic is so appealing. The idea here is that we take an antibody. Here you can see a nice cartoon, the two heavy chains, the two light chains, and we have that beautiful Y shape that we associate with an antibody. But what we're going to do is use the specificity of an antibody so that it only goes to specific areas, and in most cases it's targeting the cancer cell itself. And then what we do is we attach the payload, that cytotoxic drug, whatever anti-cancer or whatever therapeutic you're developing, but that payload or warhead, I've heard both of those terms, but it's attached via a linker to this antibody. And so the idea is, instead of delivering this very toxic drug to the entire body, we're just delivering it to the target tissue. So if you haven't seen this paper, and if you're in this work, I think you probably have seen it for sure, and this is nothing new. But just in case you haven't, I found this. It was such a well-written review article on this entire topic. They cover everything start to finish with ADCs. It's really beautifully done. But they lay out the argument in the first paragraph that I've copied here. They talk about how toxic, and we know this just empirically, right? How cytotoxic, or how toxic these anti-cancer drugs are. If somebody is struggling with this in their life and they have to go to chemotherapy, it's really a tough decision because the treatments are so awful. And I've seen this in my own personal life. They're so awful that the debate becomes which is worse, the effects of the cancer or the treatment. Now, obviously, in most cases, you take the treatment because the cancer is far worse. But the goal that I see with ADC work is to try to fix this problem, right? So instead of giving that very toxic small molecule drug and have it go through the liver and all the tissues that aren't affected by the cancer and just have devastating effects across the entire biological system, the goal with the ADCs is that we attach it to that very specific delivery mechanism of the monoclonal antibodies, and with the goal of just delivering that very toxic chemotherapeutic drug just to the cancer cells itself, dramatically reducing all these unwanted secondary, issues that come up with these treatments. So I am very excited about this work. There's so much potential in it, and I love to talk to all my scientists in this field. I just want to be a sponge to soak up everything that they know. It's really amazing. So this was a graph that I pulled, a figure 4 from that article, and I thought it was really nice because it talks about the different ways that ADCs have their mechanism. Just in general, the idea is that antibody, has a specific target to the cancer cell where it's going to deliver that cytotoxic small molecule drug to have its effect to cause apoptosis in that cancer cell, leaving the good healthy cells nearby intact. And this can happen in several different ways, and there's a lot of strategies. I want to first of all say I am nowhere near an expert on this topic. There are much smarter people than me who would probably pick my argument apart here. But I do have some basic understanding, and for the purposes of this presentation, I think it's important to talk about this. The upper right graph is showing kind of how these are used most frequently, according to this paper. You have your ADC, so you have your antibody-drug conjugate targeting the landing on the target cell where it's brought into an endosome. The idea is that small molecule drug is then able to be released at that point, where it can either have effects on, if it's an antimitotic, it might be disrupting the microtubule formation so that that cell can no longer divide, or it could be having a targeting the DNA itself to cause some effector there. The antibody is recycled through normal mechanisms, so very little concern as far as some kind of result from the antibody itself. And then, hopefully disrupt the growth of that tumor or kill the cell itself. That's a general idea. And then there's another strategy in the lower left box where through various strategies, we could be actually trying to stimulate or encourage the body's natural mechanisms to degrade that cancer cell, whether it's through a macrophage or some other, immune reaction to help that your body's own immune system to become the mechanism for destruction of that cancer cell. So that's another very powerful strategy that's being used. And then these can also be used as disruptors. So in the lower right panel, what we see is an antibody that's being used to disrupt some downstream effector that's causing a cancer issue there. So lots of different strategies. Again, I am definitely not an expert, and I would love to hear some experts add their input on this and to give us some more education on this particular topic. I thought this was just kind of funny. This is a little fun fact slide here. So I don't know how many times I'm, watching TV and a commercial comes on, and then they show the drug in the commercial and it ends in MAB. And I'm like, oh, that's a monoclonal antibody drug. That's one of those large molecule therapeutics. And certainly ADCs are going to fit in that category. Here is just a few. So anytime you're watching a commercial and you see something that ends in a MAB, you're going to know that that's a monoclonal antibody drug. All right, so let's talk about what types of analyses are being done in this field. The first one that I've listed here is basically, how do you develop that antibody to target that specific cancer cell that you're trying to reach with this therapeutic? I've seen groups that are working with natural antibodies, so they need to purify their target from a mixture. That's one type of thing. We also have people that are making their own antibodies, so they're synthesizing monoclonal antibodies. Now, you could be synthesizing different heavy chains and different light chains and attaching them and mixing and matching. We've seen a lot of different strategies, but this is definitely one major field of study in ADC work that I've come across. Another one is the conjugation reaction efficiency. So how, and this is probably a little more on the manufacturing side, right? If you're making a therapeutic, you want to have a cost-effective, high-efficiency process to develop your ADC. What reaction do I do to make the antibody that I want to use? How do I attach the heavies and light chains together? And then, of course, what process and what linker do I use to attach my payload to the antibody? And then if every time I run that reaction, I only get a half conjugation, so I still have a whole bunch of small molecule drug left over in my reaction chamber, and then I only have a small percentage of that product being my target, we want to improve that efficiency. So we've seen quite a bit of work in this particular area. And then, of course, as with any therapeutic, we have DMPK. We want to look at what is the best dose to use for a particular treatment. How well does that achieve the goal of killing the cell, the target? What effects are going to be in the body as far as liver toxicity, how is the drug delivered? We want to see, all the issues related to that final output of the therapeutic. How well is this therapeutic working? We want to characterize all areas of that. And then one that we still do see, is the raw material testing. So some people are receiving their antibodies from a third party, or they could be receiving their surfactant from a third party, or any number of the materials that they're going to be using. They want to ensure the quality of that they're receiving, or they could be, even if they make everything in-house, they want to ensure that they're sending that product to the next step in their chain at a consistent level. So this is another area that we see in this. I am 100% sure that I am missing many other things. So these are the main ones that I've seen, but if you know of others, please let me know. I'm always looking to learn more. So let's talk about the chromatography that's used in these types of analyses. They really fall into two main categories. And for the purification work, this is almost always done with size exclusion. There obviously is reverse phase purification that can be used, but it's not, as far as what I know, it's not used so much in ADC work. Why is that? Because when you use reverse phase and you're trying to collect a fraction, under reverse phase conditions, that protein becomes denatured and it's no longer biologically functioning. So that's why size exclusion is really the dominant analysis here. Because with size exclusion, you can use all aqueous mobile phases, so you're not denaturing that protein. When you collect your fraction, you have a biologically active product that you can do further testing on, or whatever you would like to do with that. The other, for analytical chemistry, reverse phase is still used quite a lot for this field as far as I can see. Most common column that we see, especially for intact antibody analysis, is a wide pore, so 300 angstrom pore size C4. That's your quote-unquote protein column. And it works well, obviously, or it wouldn't be used so widely. But I'd like to talk about why that may not be the best solution there. The other kind of main area that we see with reverse phase analyses in this field is if you're trying to characterize that surfactant, or if you want to look at that unbound small molecule in your, your conjugation reaction. Anytime that you want to be able to see what's happening, because there are still small molecules in your protein mixture, you still may have a need to characterize them for some purpose. In almost every case, what we see the solution that's being used is you first do a protein precipitation, you collect the supernatant, and then you do an analysis reverse phase chromatography on a standard pore size C18. So we want to talk about why this may not be the best solution and what we think is better and what we have for that. All right, so here are the challenges that we see. So with size exclusion, the main challenge we see is even still to this day, a lot of size exclusion is done on a polymer-based columns. They're usually large particles, polymer-based, and why is that an issue? Well, polymer-based particles, they're squishy, right? They're flexible. So if you're using any kind of high flow rate, that bed can compress, you can get channels, it really can disrupt and reduce the efficiency of that column. The other main issue with that is when you have polymer-based particles, if you change the mobile phase, so you're switching from one solvent, one solution to another, that can affect how much swell those particles are going to have. So that's not ideal. So not only do we have all these like mechanical issues, but it just isn't high efficiency. We all know that. If you're doing size exclusion, you really don't expect to have very high resolution power with that SEC. You're doing very crude separations where you have orders of magnitude of difference in size, and it functions fine and we make it work. But we developed, and this is under the solution heading, we developed a size exclusion column where we tried to fix a lot of these issues. We first of all use a silica-based particle. So much higher resolution. It's a small particle, it's 3 micron. We designed it from the beginning to be able to be used with mass spec. So that really ups the resolution factor for SEC work. So we really have a competitive product in that space. For reverse phase, I had said that everybody's using that wide pore C4. Well, and it works great, right? Everybody's using it, it's fine. But I'd like to introduce our product, which is Presto FFC18. This is a non-porous particle. It has no pores. So this has a lot of advantages for that intact antibody analysis, and we're going to take a look at that. And finally, we had talked about when you're trying to characterize a small molecule in your protein mixture, we've got to do that protein precipitation first. How would you like to get rid of that? I know if I were doing this, I definitely would. And we have a column called Cadenza HS18 that allows you to do that, and we're going to get into that in more detail. All right, so first, let's talk about this SEC. Our column is called the Entrada SEC. And I like to say this is a high efficiency silica-based SEC for MS. Now, there's a lot of packed into that, and let's get into why that's the case. We had already talked about some of the challenges that we saw, right? A lot of C18 or SEC columns are polymer-based, they're squishy, swells with solvent. They don't use volatile salts, right? They might use sodium chloride. So all these things are just, you use what you have and what's available to you. But when we said we were going to make an SEC column, we wanted to fix all these things. And I feel like we have. Our column is, as I said, silica-based. And here I'm showing it being used. This is actually from one of our customers in Japan at Nagoya University, Dr. Kuwata. Excuse me. And what we're seeing here is some recombinant HSA. But look at the detail. When you're using mass spec, now all of a sudden we're not resolve to the dark ages of SEC here. This is where you can do SEC and have high resolution. It's really, really powerful. So, and you can see in the mobile phases, we're using an all aqueous ammonium formate mixture. So you're not going to denature those proteins. It's really a fantastic solution here. Here I'm showing monoclonal antibodies, some IgGs. So here we are perfectly in that 150 kilodalton size range for ADCs. I like to show this. This was feedback from one of our customers, and this was very early when we introduced this column. And we worked with this gentleman from Genentech, and he sent us this email completely unsolicited. But he basically, in a nutshell, said, TSK gel, they're a big name in SEC. They were using an SW3000, which is a 2 micron particle size silica base, so very similar to ours. But his impression was that it was not better. He got one big fat peak about 5 minutes wide, and ours worked much better for him. In fact, this is the one they went with. One thing to note, the column he was using, there's 2 different lengths here, and you can see the price is very high. TSK Gel can do that because they pretty much own the SEC market for the most part. But coming into this, we put a whole lot of technology at a very competitive price point. All right, so just to sum up, we use a silica-based particle. It's a 3 micron. We have wide solvent compatibility. It's designed from the beginning to be worked with mass spec, and it's not going to break the bank. So if you're doing SEC work, that purification work for your ADCs, please let me know. We can set up a trial basis and see if our SEC column is going to be a good solution for you there. All right, so let's move on to the reverse phase analysis. The first reverse phase analysis issue, the challenge here in the chromatography work. So I said, that standard protein column is what we see being used the most for intact antibody analysis. But you have to think, even 300 angstrom pore size or 450 angstrom I've seen, or even 1,000 angstrom pore size, the size of an intact antibody is almost half that. So it really prevents high resolution analysis. And I'm gonna show you more detail on that, and why our non-porous Presto is a better solution. So let's talk about Presto. Here's a nice SEM. You can see the particle itself. This is a solid silica. There are no pores. And with that, there are so many advantages for this type of work. Not having any pores means you have no size limit. We've analyzed with very sharp peaks, molecules up to like 30 million Daltons. Can you even imagine that? So certainly is going to work very well for ADC work. Another thing that people may not think about all the time is that when you introduce large pores to a silica particle, it makes that particle very brittle. It's easy to crush. Certainly 300 angstrom pore size is in that boat. But you get to larger pore size, like 450 angstroms or 1,000 angstroms, that particle is going to be very delicate. That's a huge advantage for the Presto. Being solid silica, this thing is almost indestructible, and we have no pores to trap things. So these columns last for a very long time. So what do we use this for? Really, as I said in that first column selectivity chart slide, any molecule over 70 kilodaltons, we're going to gravitate towards using the Presto. Now, you might say, well, I would never use a C18 for my protein work. I always use a C4. And you're absolutely right. By standard convention, that is absolutely the case. But you have to consider, with Presto, we have no pores. So the surface area is dramatically reduced. We're only bonding stationary phase to that outer circumference of that sphere. So in all practical terms, this behaves as we would see on a C4. So we're not getting extreme retention for proteins. It actually is a very nice package for this type of analysis. All right, so let's take a look at this comparison that I've kind of introduced, that wide pore C4, and why it may not be the best choice for intact antibody analysis. Anytime I've talked to a scientist in this field, you ask them the size of what they're working on, they're almost always going to say 150 kilodaltons. You've got two heavy chains that are about 50 each, the light chains are 25 each, totaling a 150. And that's the convention. Everybody knows that. I think what people, at least from what I've heard, what they don't talk about is its actual dimensions. These intact antibodies are about 150 angstroms wide. They're about 160 angstroms tall. So now I think you can start to see why, even at a 300 angstrom pore size, this might not be the best strategy for an intact antibody analysis. So, as I said, we have a wide pore C4. It's called Entrada WPRP. And when you look at that 300 angstrom pore size, which I've shown here, kind of in the center, you can see that it really doesn't take very many antibodies to where you're going to start having a lot of collisions. It's going to collide with the structure of the silica, the walls. It's going to collide with other antibodies. And why do we care about this? Well, in chromatography, when you're looking at high efficiency, you care about mass transfer. How does your analyte transfer into the stationary phase and cycle out of the stationary phase, move forward in the column, and then cycle back into the stationary phase and does this sigmoidal like cycling into and out of the stationary phase? And you want that to occur as smoothly as possible. You only want the analytes to have an interaction with the column, with the stationary phase. But when they're exiting the stationary phase and when they get into that pore space and they're interacting and they're bumping into a bunch of stuff like other antibodies, well, this can disrupt that smooth mass transfer and give you a lower resolution. It's not as high, not as sharp a peak shape because you have all these disruption events. And that's really the main reason why these wide pore columns are not best for such large molecules as intact antibodies. But it still works. That's why everybody's using it, right? And here is our in wide pore C4. This is Entrada WPRP. Here we're looking at intact IgG2. This is work by Dr. Dillon at Amgen. And you can see, if you're in this field, you would probably look at that and you would say, that's a beautiful peak. Look at that. It's nice Gaussian shape, very symmetrical. Like, that's a good chromatography for this field for the most part. But as I said, because we have all those disruptors to smooth mass transfer, it's not as efficient as it could be. So let's take a look at that Presto. As I said, this is that non-porous 2-micron particle size. And yes, it's a C18, but it works great for proteins because we have that overall lower carbon load to a traditional C18. All right, you can see our mobile phases, very simple. This is work by Kumiko Sakai Kato. This is, they're based in Japan's NIH, their National Institute of Health. This is a wonderful reference paper. They do a lot of method development work and they show a lot of that work. Like they tested the best temperature, a lot of factors. So it's really another very good paper to read. But if you notice here, let's take a look at the chromatography. So this is the same molecule, right? This is an IgG2. And This is IgG2. And if we were to put a line that connected the tops of all these little peaks here, we would have essentially the same trace that we saw on that wide pore C4, that nice Gaussian curve, that we would see and we would say, oh, that's a beautiful symmetrical peak. But what do you get with Presto? Look at how much more separation you have here. This is vastly different, vastly better, not a little bit, it is dramatically better. So this is what I'm talking about as Presto being a much, much higher resolution for very large molecules like these intact antibodies. Okay, so as I said, here's an analysis with a 30 million Dalton double-stranded DNA from salmon. Nice sharp peak, so huge advantage there for Presto. And as I said, this really opens up that large molecule space to high resolution reverse phase. Before, if we're stuck with wide pore C4s for all of our large molecule work, it has some limitation. This column really doesn't have that limitation, and it opens up that entire space to high resolution chromatography. Here's another example, 450 kilodaltons. Whenever I introduce Presto to people, I want to tell them that non-porous works great, has so many advantages, but it doesn't operate exactly the same as fully porous material does. And one of these to pay attention to is what's shown here on this slide. What we're seeing is, we start with an initial 6% B, which most people, that's not very much. But you can see that it does affect the chromatography. Presto is a little more sensitive to this effect than most other columns. But you can see once we get to starting with 0% B, our peaks are dramatically improved here. And so these last four traces here are showing starting with 0% B, and then we get beautiful peaks here and different. We're testing different gradient slopes here. Here's a very shallow slope. Here's a more steep slope. Why is that? Well, every column shows this. Every single reverse phase column will show this. It's called peak focusing. So imagine you introduce your sample into your injection loop. It's traveling through the tubing to the head of that column. There is some dispersion that's going to occur of your sample in that process. If you have B, mobile phase B, which is your eluent, right? If you have that already present from the beginning, then when your sample reaches the head of the column, it's moving, right? So the first analytes that reach the column are already moving before the tail end of your sample reaches the head of the column. So there's some introduction of band broadening there. When you start with very low B, or in this case 0% B, what happens is when that bolus of injectant reaches the head of the column, it stops. It sits right down. And it allows the tail end of that peak bolus to condense itself and catch up with the front part of that bolus so that you start with a very sharp peak. And all columns show this effect, but Presto is a little more sensitive. So that's something to consider. I also mentioned the steepness of the gradient. This is really analyte dependent. I've seen on Presto some analytes prefer very shallow gradients. In this case, we saw the steeper gradient had that much sharper peak. So Presto, with a few small changes in your gradient or mobile phase constitution, you can go from a peak shape like this to a peak shape like this. And again, we're using a 1 million Dalton solution here of hyaluronic acid. So pretty cool stuff. The other thing I like to point out is this is a 4.6 millimeter ID column, and we're using 0.5 mils per minute flow rate. Now, most commonly, if you're using 4.6 millimeter ID, column, you're using about a 1 mil per minute. So you can see that we're using roughly, excuse me, roughly half of the flow rate. Now this is actually very important when using Presto. We don't have the pores to slow down the mobile phase and have that intimate interaction between analyte and stationary phase. Everything is occurring on that outer circumference. So because of that, if we were to use 1 mil per minute on this, our retention would go way down because we're just basically super freeway flowing analytes right past the stationary phase. So we do recommend reducing the flow rate to help promote good interaction with the stationary phase. So those are some of the minor things to consider when developing a method on Presto. You can always reach out to me anytime. I've been doing this for over a decade, so I have a lot of experience. I can definitely help you dial it in to where you want to get it. Here's kind of a side note. If you're using mass spec, hey, using a lower flow rate is pretty nice. In this case, you could probably even use a 4.6 millimeter ID on your mass spec and still have it work great. All right, here's something else to show. It also happens to be our best column for peptide mapping. Now, I wouldn't intuitively have thought it is, but when we tested it out, it had more than twice the peaks. So it actually works very good for peptide mapping. You can see how much taller the peaks are on the Presto column versus a standard pore size C18. So if you're doing peptide mapping work, Presto might also be good for that. All right, so now let's talk about this last section, and that's where you want to look at, you're more concerned with, or you're being asked to characterize the small molecule in your protein mixture. As I had said, this almost always requires some protein precipitation, and then the rest of the analysis is done on any old regular C18. Well, we think using our direct injection column is a better option. So let's talk about this. It's called the Cadenza HSC18, and I absolutely love this column. This really speaks to Yazawa's mantra about trying to provide solutions to chromatographers that aren't out there. I've seen all kinds of direct injection columns out there. Monolithic columns, that meso megapores. We've seen a lot, but they so far, all the feedback that I've heard from people is that they just do not work. They don't hold up. And I love Yazawa's solution to this problem because it's so elegant, so simple, and it works. Basically, his solution was, so he started with our Cadenza C18, which, first of all, a little side note here, is a very good C18. It has high steric selectivity, good isomer separation power. It's a very good C18. What he did was he started with that Cadenza C18, and he added one extra ligand. What this ligand is very long. It's actually much longer than what's shown in this cartoon here. And it's polar but non-ionic. So we're not going to have ionic ion exchange interaction. But what it's going to do is act as a repulsive force to push the proteins out, unretained, off the column, while allowing the small molecules. The small molecules don't care about a polar force field, right? They're going to go right through that, be retained by the C18, and in essence, function like any C18 that you've used. We're just going to exclude the proteins. So here's a little cartoon that I made to show this. And I've shown the particle. It's actually a standard pore size, fully porous 3-micron particle. But these very long ligands, so you can see the dark black line is representing that C18 film that's on the surface of that particle. But remember I said those polar ligands are much longer? So in essence, what that does is create this force field around the particle. So in the cartoon I've made here, the large red dots are proteins. The small black ones are your small molecules. And those proteins are going to hit that force field, bounce off. They're not even going to go into the pores and interact in any meaningful way with the C18 or any of that. They're just going to be pushed out in that early part of the run, unretained, and release off the column. While those small molecules, which don't care about polar force fields, they're going to go right through and be retained by the C18 and have that standard small molecule analysis that you like. Very simple. It's so beautiful. So let's take a look at what that looks like. When we first developed this column, we wanted to fully characterize it. You can see this bottom trace, this baseline. We had some small molecules in that mixture. And what we did was we mixed this standard material in with larger and larger, peptide and protein mixtures. And what we found was right around 30 kilodaltons we started to see this kind of exclusion occurring. So certainly, and we tested this up to 670 with thyroglobulin, right, with 670 kilodaltons. And look at this, it's a beautiful trace. We get that nice sharp peak of exclusion, and we have our standard small molecules in here being retained beautifully. So certainly it's right in the wheelhouse to work with ADCs for this column. Here's another example. We had these standards with three peaks here. We mixed it into some bovine serum, and you can see we have no shift in retention for our small molecules, but we're getting that large protein exclusion peak right out here. So you can see very standard kind of mobile phases. In this particular example, we've added some ammonium acetate just to make sure we don't get any tailing of this early protein peak. But other than that, it's really just a simple acetonitrile aqueous and acetonitrile organic gradient that we'd use with any C18. So you don't have to do a lot of special things to get all the benefits out of this cadenza HS C18. And another thing I'd like to point out is that our analytes are coming out, within this early window where we're still under high aqueous conditions. So all those proteins are moving undenatured through the column and exiting the column before we get to higher organic levels that could potentially denature the proteins. So this is really an elegant, beautiful solution to this problem. What if you're using mass spec? Well, this is also pretty simple. You just use a switching valve to run the proteins are coming out in a predictable way, right? They're coming out very early, so we can send that early flow to waste and put it back online for our small molecules as they come through. This was kind of nice. This group is based out of Korea's Institute of Science and Technology. They were doing 100 samples per day, and they said that they got over 1,000 samples on this column. So very nice to hear feedback of good lifetime for this type of work on this column. So when are you going to use cadenza HS? Anytime that you need to do a protein precept before analysis, cadenza HS is a good candidate to potentially put into that work. Even with small molecule drug discovery work, people are using this. If you have 1,000 samples to test for one metabolic change, you're doing protein precip on all those to analyze them by standard C18, right? With this, you don't need to do that. You can just put all that on the C18 and it'll work great. Certainly with ADCs, we're seeing it. Sometimes people want to characterize the unbound payload small molecule drug in their ADC mixture, right? Or you might be trying to characterize that surfactant material. So in any of those cases, this column is a great option for that. So let's take a look at, sure, it makes it easier, right? I don't have to protein precip. That saves me steps, it saves me time, it saves me materials, it saves me vials, right? But another thing to consider is that, like, let's take, for example, you're doing surfactants. You need to characterize the surfactants in your protein mixture. When you're doing a protein precipitation step, you have to control for all the variables introduced by that precipitation step. When you're using cadenza HS, you don't have to, right? We're not molesting that surfactant or the small molecule in that sample in any way. We're starting and using that raw sample. So even I would argue the data is stronger when using HS18. So that's another very good reason to consider that. Here's an example showing that surfactant analysis. You can see all of our antibodies are coming out unretained. And then we have our tween 20 in this particular example. And we have a beautiful linearity curve, right? So this speaks to consistency and just how well it's used. And guess what? I found another paper that talks about this. This is from another group at Amgen, I believe. I would definitely look up this paper if you're doing that work, if you haven't seen it already. But they also made a poster that they presented a lot of this data at HPLC in 2010. And in this wonderful work, they tested several different columns that are direct injection column, and they chose the cadenza HS over all these for a variety of reasons. I just want to go through some more of the details. So, they talk about the challenge in working with these types of samples, right? You have to protein precipitate, but they wanted to be able to analyze these without doing any protein precipitation, and that was kind of the impetus for this work. And it was wonderful that our column was the one that came out on top on this. You can see in the first trace there, you see the proteins coming out with their surfactant showing up. When they did a blank, there was no carryover, so that's really nice. It's a great work because it also did a lot of work showing their method development process. And they tried several different gradients. You can see there, gradient 3 was a very good one. They also did that linearity and also found a very good consistency. So in their conclusion, I wanted to point out their mobile phase, right? Very simple mobile phases here. Nothing exotic. So that's another beauty of the HS. You can use it like you would any other C18. It just has this magical character of being able to push out the proteins. So we're not having to do any kind of weird things with it. But their conclusion, a simple and sensitive method for determination of PF68 was developed using the cadenza HSC18 column and ELSD detection. The results of specificity, linearity, precision, which are repeatability and accuracy, limit of quantification and robustness, showed that the method was suitable for testing PF68 clearance in MAB preparations. So again, this column has so many uses in the ADC world. All right, so just to kind of put a wrap on this, I apologize for going quite long on this, but it's a big topic. MTACT, we have some really, really unique columns that provide solutions for researchers in the ADC space. We've gone over how our SEC column gives you so many advantages over what's out there in the market today. It's MS compatible, wide solvent compatibility. It can be used in GPC or GFC, and it's a very competitive price, right? Our Presto FFC18, that's that 2 micron non-porous particle. It really opens up large molecule space to high resolution reverse phase. It is so uniquely suited for this work. I guarantee you're going to love it. And then, of course, our direct injection, that cadenza HSC18 column. This is such a workhorse. It has so many good areas of applicability, right? If you're looking to try to characterize the surfactant clearance, if you're looking at how well your small molecule cytotoxic drug is attaching, is conjugating to your antibody, there's so many uses for this column. But again, if you're doing protein precipitation and you would like to eliminate that from your steps, then cadenza HS might be a great solution for that.
Well, thanks for watching. I hope that it was helpful. This is, again, such an important field, but the chromatography can be quite challenging, especially since we're dealing with molecules that are 150 kilodaltons or even larger. I hope now you understand that we make some great products that can definitely be a benefit to you in your lab. Our Entrada SEC for high resolution MS SEC work. We also have the Presto FFC18, that 2 micron non-porous column that has no size limit. It can easily handle 150 kilodalton antibody. And of course, our cadenza HSC18, that wonderful direct injection column. It's very reproducible. Whether you're trying to do protein depletion work to uncover what's going on with the surfactant or the unbound small molecule, that's a fantastic product. If you have questions about this or anything else, please hit me up. I'm at info@mtacusa.com, and I'd be happy to work with you.